My name is Todd Lowry. I'm a Distinguished Research Fellow at Academia Sinica in the Institute of Biological Chemistry, and I also have an appointment at IBS at National Taiwan University. So I am originally a native of the United States. I received my undergraduate degree in the late 1980s in the University of Montana. I did my PhD then at the University of Alberta in Canada, and then started my career at Ohio State University, where I was an assistant professor for 7 years and then received tenure. I moved back to Canada after that time and spent the next 17 years at the University of Alberta, and then in 2019 relocated to Taiwan. So my laboratory focuses on the role of carbohydrates in biology, and we are particularly interested in understanding and making compounds that can be used to probe the function of carbohydrates in biology. And so my group spends most of their time doing chemistry to make molecules. And so we do this by starting with starting materials. We carry out a series of reactions. We make typically molecules that are called oligosaccharides. So oligosaccharides are molecules that are found in nature and that play a number of important biological roles. So my group is particularly interested in trying to understand the role of glycans or carbohydrates in bacterial disease. And one of the major focuses of our group over the last 15 or 20 years has been on glycans from a family of bacteria called mycobacteria. So many of you may have probably familiar with one mycobacterium. The most famous mycobacterium is Mycobacterium tuberculosis, which causes the disease tuberculosis. There are a number of other families of this bacteria. Another is called Mycobacterium leprae, which causes the disease leprosy. And many people who have suppressed immune systems, for example, people on immunosuppressive drugs, or people, for example, who have HIV, also are susceptible to these types of infections. So in many places we think about tuberculosis as being a solved disease, but actually it's not. It remains a threat throughout the world. About 1 million people die every year from tuberculosis, and it's particularly a problem in the developing world, parts of Asia, Africa, and some places in South America. So my group has been interested in trying to understand how this organism, how the carbohydrates in this organism allow it to survive. We want to understand how these molecules are put together. And the idea there is that if we can understand that process, we could then design molecules that could inhibit that process. And these molecules then could be potentially lead for new drugs to treat tuberculosis. There's a lot of interest in developing new tuberculosis drugs because of drug resistance. And so, for example, increasingly we find that many mycobacterial infections are resistant to one, two, or even in some cases all of the existing antimycobacterial drugs. So this is a serious problem. In addition, these carbohydrates in the mycobacteria interact with the host immune system, so for example the human immune system, and they confuse it basically. And so we're trying to understand how these carbohydrate structures interact with the immune system, and can we then use those types of interactions or those information to then, for example, develop better vaccines or develop better diagnostics that can be used to detect TB. And again, we think about diagnostics as being a sort of solved problem, in particular with regard to TB, but there are limitations in the existing diagnostics that are available for tuberculosis. And so there's a lot of interest in developing new drugs and new diagnostics. And so one of the collaborations we have at the moment that I'm particularly excited about is a collaboration with a group in Europe called the Foundation for Innovative New Diagnostics, or FIND. And we've been working with FIND over the last 3 to 5 years to try to develop antibodies that could be used to develop better diagnostics for TB. So that's a major focus of our group is mycobacterial disease. We're interested in other types of carbohydrates as well, from bacteria. One of the other families of bacteria we're interested in are Campylobacter. And so you probably all had a Campylobacter infection. If you've ever had food poisoning, there's a pretty good chance that what you had was Campylobacter. You don't hear a lot about it in the news. You hear a lot more about Salmonella, Listeria, or even E. coli. And the reason for that is that Campylobacter isn't so serious in the sense that you don't feel well for a few days, but then you recover, usually without any problems. But the major cause of traveler's diarrhea, for example, is Campylobacter. And so we're interested in trying to understand, again, how the carbohydrates in Campylobacter interact with the immune system, and then try to use that information as a way of designing better diagnostics. And also vaccines. So again, the group focuses on the synthesis of these Campylobacter glycans, and then we collaborate with other groups to do some of the downstream biology. So the third area that we spend a lot of time working in now is in the area of viruses. And of course, in the news recently, we've heard a lot about viruses. The ones we work on are actually not important human pathogens, but they're actually a family of interesting viruses that interact that infect algae. Okay. So if you've had a bit of biology or biochemistry, you know the way that a virus functions is it goes in and it incorporates the host genome into its own genome, and then it uses the host genome to make its structures. And so that's certainly true. So viruses, for example, like HIV or coronavirus, will produce molecules that contain carbohydrates. But the way they do that is they go in and they take the host biosynthetic machinery and they use that to make their own glycans. The viruses we're interested in, chlorella viruses, are very different than that in that they have their own family of enzymes that produce carbohydrates, and this is interesting. So why have these viruses evolved to produce these different glycans? Why have they evolved to have their own carbohydrate processing machinery? And so again, the molecules we're making here are being used to try to answer the question, how are these put together? Why does it do this? There's not a lot of health applications to this, but it's an interesting problem nevertheless. So in terms of how we choose to work on problems, I've always been fascinated by unique structures. And so if I look at a molecule or look at a biological system and see a molecule that looks cool, frankly, looks interesting, I'm immediately attracted to it. And so a lot of what we do really starts with just being intrigued by an interesting structure. Ideally then we try to find a way in which we can convert that sort of interest in a structure into something meaningful from a biological perspective. And so that's the types of problems we work on do almost always have some sort of important biological relevance, although that's not a requirement for everything. We're really more interested in trying to understand these cool structures and also how they're produced and how the body interacts with them. Okay. As far as the expertise in my group, most of the students in my group spend their time using organic chemistry to make molecules. We also use spectroscopic methods to characterize the molecules, and then also we do some biochemistry in terms of protein expression, protein mutagenesis, binding assays, enzyme assays. And so students in my group get a fairly broad range of training from what I would say is pretty traditional organic chemistry all the way through to biochemistry. We also collaborate a lot with people. These are immunologists, structural biologists, biochemists, microbiologists, and whenever we have the opportunity of working with somebody working on an interesting problem, we try to do that. And the collaborations I have are with people here at Academia Sinica. We also have collaborations in Canada and the United States, as well as a number of places in Europe. So as far as my approach to mentorship, I like to work on fun problems, and I like to have fun. And so basically, if something isn't fun, we shouldn't be doing it. And that's really the way I feel about research. And so what I'm looking for is for interesting people who are really interested in the projects that we're working on. As far as a boss goes, I'm not a terribly hands-on kind of guy. I like students to gain independence. I'm certainly available to help you whenever you want, but I really encourage you to become independent and try to test out your own ideas. And so what I like to see are keen, motivated students who want to take on a challenge. I like to work with students. It's one of the reasons I do this job. Again, as I mentioned, everything has to be fun. So if it's not fun, then we shouldn't be doing it.